So we are interested in exploring the function of the inner lining of the gut, the cells called intestinal epithelium, and they've been long suggested to potentially have a major role in Crohn's disease. But so far, the problem was that most of our knowledge comes from mice and animal studies until we were able now to grow these mini guts from actual patients. And this is what we've done in a fairly large number, over 300 of these little mini guts from over 160 patients. And we figured out that there are certain changes called epigenetic changes, which refers to changes that do not change the underlying gene sequence, but the way the DNA molecule is modified. And these modifications determine the function of the cell. And so, for example, they determine whether a cell is a brain cell or a gut cell. But sometimes these changes also happen when we compare cells from a patient and a healthy control or a healthy individuals. And this is what we've done in this study. So we were quite excited about this because we found that these changes occur in a subset of patients with Crohn's disease, which is really important because it could explain why not every patient with Crohn's disease has the same disease. In fact, almost every patient has a unique disease, and they differ in terms of severity, outcome, whether they do or don't respond to treatment, whether they ultimately need surgery or not. And so finding things that differ between patients with Crohn's disease as well as between patients and controls is really exciting because it explains what we see in practice. And I think then the real excitement came when we figured out that the organoids that came from patients or in which we saw the most dramatic changes, they came from patients that tend to have a more severe disease. So this is sort of the final, if you wish, confirmation that these mechanisms seem to be truly important. And then leading on from that, of course, this shows that that mechanism we figured out or we uncovered may represent a great new opportunity as a therapeutic target, as well as a way to optimize treatment, a personalized treatment.